Howdy everyone, today we're going to be looking at cellular respiration, photosynthesis, and the two ways of fermentation, right, in an animation style. So we're going to start off with cellular respiration, right? We're going to start with glycolysis, of course. We first start with the first phase of glycolysis, which is the investment phase. So we have this glucose molecule, that's 6-carbon sugar. We're gonna reduce it a little bit, temporarily reduce it. We call this oxidation of glucose, but first you gotta reduce it a little bit, right? Gotta split it apart. You need energy to do that. So you use 2 ATPs, right? And then you get your ADPs, and you're left with 2 glyceraldehyde 3-phosphates, or G3P or PGL, whatever you want to call it, right? So now we're going to take 4 ADPs and 2 inorganic phosphates. We're going to come to these, and we're going to reduce it, or we're going to oxidize the molecules itself, but the energy molecules, ADP, we're going to reduce it into ATP, right? So we net 2 ATPs because we used 2, we gained 4, that's a net of 2, right? And there's some electrons hanging out there, and NAD+, that oxidized form of that electron carrier, is going to be greedy, and it's going to be like, yummy, yummy, yummy in my tummy, gimme. So now we get 2 NADHs, right? Now we're left with 2 pyruvates, right? This is what we call the payoff phase, because in the investment phase, we use 2 ATP, but in the payoff phase, we gain 2 ATPs net, of course, and then we get 2 NADHs, right? And we're left with pyruvate. This is an oxidized form of G3P, by the way. Next comes the link reaction. In the presence of oxygen, pyruvate will move into the mitochondria. So let's see that happen there. Yep, good to go. Inside the mitochondria we are, although that was red and now this is blue. Doesn't make sense. Claire did it. Don't ask me why. Okay, moving on. Right, this is also called pyruvate oxidation. So keep those two terms synonymous in your head. Coenzyme A is going to attach to that 2 pyruvates. It's going to yeet a carbon, and it's going to release those yeeted carbons as CO2. So we gain 2 CO2 per molecule of glucose, right? And we're left with that 2 carbon chain here. And then we also have that coenzyme A attached. We call that acetyl coenzyme A. We have 2 of those, right? NAD+ then comes in and is yummy, yummy in his tummy, reduces itself into NADH. We get 2 NADH from the link reaction. Moving on. We have the citric acid cycle. So we have those 2 acetyl coenzyme A's, right? And it's going to see oxyl acetate. It's going to want to bind together. Coenzyme A leaves, and we are left with a 6-carbon citrate, right? We're going to start oxidizing this molecule, right? So we're going to take NAD+, we're going to reduce it into NADH, right? And we get 2 of the molecules there, or 2 molecules of CO2. We also gain 2 NADH, right? Same thing happens for the next carbon, right? We're going to reduce NAD+, oxidize that, right? We're going to get 4 NADHs from this process. So now we have ADP molecules and inorganic phosphate, right? We're going to reduce those molecules into ATP, right? And we're further oxidizing those 4 carbon molecules we see there, right? And we're going to take FADH, which is the oxidized form of this electron carrier. It comes up here, steals some electrons, becomes the reduced form, NADH2. So moving on, we're finally going to take NAD+. Again, we're going to take 2 of them. We're going to reduce them into NADH, right? From this total cycle, we get 6 NADH, 2 ATP, and 2 FADH2. And now we're left with oxyl acetate, which is what we started with in the fixation phase of this citric acid cycle, right? It fixated on the acetyl coenzyme A. That's going to be sent back to the beginning, where it will attach to another acetyl coenzyme A. All right, moving on, we have oxidative phosphorylation. Pretty simple here. We have some electron carriers down there in the bottom left, as you'll see. NADH goes first. It says, I'm going to oxidize myself, becomes NAD+, drops off some electrons. FADH2, remember that reduced form of that electron carrier, comes in, says, okay, I'm going to dump my electrons in this protein here. It will do that. It will become oxidized, and now we go to the next part here, which is chemiosmosis, right? This is part of oxidative phosphorylation, you could argue, right? So these electrons that are moving in these increasing electronegative proteins, right, they're going to pump H plus ions across the mitochondrial membrane into the inner membrane space. This will, of course, make a concentration gradient of high H plus in the inner membrane side and low H plus in the mitochondrial membrane. What you'll notice with electrons is that they're going to go into that cytochrome, I believe it's called cytochrome 4. Those electrons will then bind with oxygen and H plus ions, and that's going to form H2O. And that's a waste product that gets yeeted from the cell, right? Or used, or however it feels like it should use that H2O. I don't know. I'm not the cell. Moving on here, back to chemiosmosis, right? H plus is going to flow into this enzyme we call ATP synthase. Synthase meaning building, ATP, synthase, building ATP, right? H plus is going to go through it. It has a rotor in there. I don't show this here, but that rotor will start to spin, and that will create energy that helps us reduce ADP and an inorganic phosphate into that high energy ATP, right? And that H plus is returned back to the mitochondrial matrix. Moving on here, we are now going to move into photosynthesis, alrighty? First step, we're going to take our H2O, and we're going to split it in half, and we're going to get oxygen gas and a bunch of H plus ions. How fun, right? Let's look how a photosystem works, right? And just to clarify, we're in the light-dependent reaction. We'll go to the light-independent reaction or the Calvin cycle next. But let's see how a photosystem works in the light-dependent reaction. So, as you know, the sun emits photons that cells will pick up, right? But how does it do this? Well, it takes all that light, right, that photon, it's going to absorb the red, blue, and yellow light, and it's going to repel or reflect the green light. That's why plants are green, by the way, guys, if you didn't know. So that light energy that the sun gave us, that photon, is going to excite the chlorophyll pigments, right? And it's going to bounce energy through them, right? So let's watch it go through. It's going to bounce. Woo, yay. It's going through, it's going through, it's going through. Come on, you can do it, energy molecule. Energy is not matter. Forget what I said. Okay. Important step here. We split H2O. What does that do? Well, we had H plus ions, so there's electrons. Conservation of matter says, well, you know, the electrons need to go somewhere, right? It's actually going to go through here. It's going to be like, oh my God, energy. It's going to go into what we call P680. That's in the photosystem 2. In photosystem 1, we call this P700, right? And that electron gets accepted by an electron acceptor. And what you'll notice, what happens with this electron, or this pair of electrons, is it's going to move through the cytochrome complex, which all that is proteins that are higher in electronegativity. Remember, electronegativity means that wanting to attract electrons. So photosystem 2 is pretty weak in electronegativity in relativity to everything else. So those cytochromes are going to pull those electrons into here, and H plus in the stroma will get shot through into the thylakoid space, right? It gets shot into photosystem 1, and what you'll notice is we have NAD, well, it's labeled NAD plus reductase. It should be NADPH, or is it NAD plus? Whatever we call this enzyme here, NADPH plus something reductase. It's an enzyme, and it's going to take... Oh, NAD plus, welcome NAD plus into the mix here. NAD plus reductase, meaning to gain energy or gain electrons. It's going to come into this enzyme with the electrons, and we're also going to spawn a magical soup H plus ion, because why not? And it's going to combine, mix it all up. We get NADPH, which is the reduced form of that electron carrier. So we're going to gain 1 NADPH per cycle here. More chemiosmosis, because why not? ATP synthase, right? Matter has a tendency to want to move from high to low concentrations. It just so happens the thylakoid space, through that splitting of the H2O, making those H plus ions, and the electrons pulling those H plus ions into the thylakoid space of the cytochromes, is going to cause there to be that buildup in that high concentration of H plus, right? It's going to move through ATP synthase, right? It's going to spin that rotor just like we saw in cellular respiration. It's going to come together with ADP and inorganic phosphate, and it's going to reduce it into ATP. Pretty simple here, guys. Moving on. Now we have the Calvin cycle, right? We looked at the light-dependent reaction. Let's look at the light-independent reaction, or how we call it, the Calvin cycle. So we start with 3 CO2s, right? And we have 3 RUBPs, right? They're going to come together in this enzyme called Ribisco, right? And it's going to give us 6 3-PGAs, right? And what you're going to see here, well, we're going to center it because centering is a good thing. Get rid of Ribisco. Goodbye. We don't need it anymore. We're going to take 6 ATPs. Quite a lot. It's not very efficient photosynthesis, which is something to keep in the back of your head. These cellular processes are not very efficient, and it makes you wonder why in evolution and natural selection we didn't favor organisms that were more efficient in their metabolic processes. You know, that's something to look into. All right, so we take 6 ATPs, and we're going to come here. We're gonna boom. We're gonna first reduce these molecules, right? Because we're reducing to build glucose. You need to make a high energy molecule. So you're going from a more oxidized, more boring molecule, and you're gonna build it up into glucose using the oxidation of ATP. So now we get ADP, right? That's the oxidized form. When you oxidize something, you have to reduce something, or in other words, when you give something energy, something has to lose. And that's what happens with ATP. So we use 6 ATPs. We get 6 of the 1,3-biophosphoglycerate. I think that's how you say that. Don't quote me on that if I said that completely wrong. Moving on, we now have NADPH, right? Remember, we also made that with the reductase, right? It's going to be all happy. We have 6 of them. We have 6 of them, right? They want to oxidize themselves. So what they do is they come to this. They're going to reduce that 1,3-biophosphoglycerates, right? It's going to reduce all 6 of them, but it's going to oxidize itself. So now we use 6 NADPHs. Those become the oxidized form NADP+, right? And we get 6 G3Ps, which should look familiar because that is 1/2 of a glucose molecule. But something interesting happens here. We can only net one of these, right? So we're going to shove that off to the side for now. This is now called the regeneration phase, right? I should have pointed this out earlier. When the Ribisco makes the molecules originally, we call that the fixation phase. Then we have the reduction phase. Quite fitting because, you know, we're reducing to make glucose, right? But now we have to regenerate, because it's a cycle, right? We have to regenerate those fixating molecules, that RUBP that we once used here. So we have that 6 G3Ps, right? We're going to use some ATP. We're going to oxidize the ATP and reduce these molecules. And what you'll notice is we get 3 of the RUBPs. And that's sent back to the beginning of the cycle, right? It's going to fixate to those 3 carbons, it's going to reduce, and it's going to regenerate. So it's that constant cycle. All right, moving on. And let's say we do it again, right? I should have also noted, right, something important to know is we use 9 ATPs in total and 6 NADPHs. Exactly what I meant by this being extremely inefficient. You know, I bet you there's some chemical way that someone has discovered that you could potentially make this way more efficient. I don't know the exact efficiency rate, but that is quite terrible. You need to go through the cycle again because G3P is only 1/2 glucose, right? So in total, for 1 molecule of glucose, we use 18 ATP and 12 NADPHs, right? So now we have these 2 G3Ps, right? What are we going to do with it? Well, we're going to combine them, and they're going to form glucose. That's exactly the cycle that's going to then be used into cellular respiration. So this glucose is going to get eaten, and it's going to be broken down by the cytosol, right, and wherever it gets used, right? And that's kind of that interdependence that we see, right? The photosynthesizers are very dependent on cellular respirators, and these processes depend on each other, right? The breaking down of glucose to get energy, but you also need to build the glucose up so you can even do cellular respiration. Kind of a weird tangent to go on, but it's good to know. Now it's time for fermentation. Feast your eyes on this beautiful kitty cat with bread on its head. Why bread? Because fermentation, or at least in alcohol fermentation, as we know, releases CO2 so your bread can rise. I thought it was a relevant image, so I thought I'd throw it in there. We're going to start with lactic acid fermentation. We go through the glycolysis the exact same, right? We're going to use that 2 ATP to split the glucose into 2 G3Ps, right? Oxidize the ATP, boom, goes over there with the 2 G3Ps. Now let's get the 4 ADP, right? We're gonna reduce the ADPs back into ATP, oxidizing this molecule further, right? And we're gonna take NAD plus, right? We're gonna reduce it into NADH, right? And we are gonna net, right? We net that 2 ATP, right? That's the important part. In fermentation, you're gonna get 2 ATP. You're not gonna get any more. You'll see that in a second. Now we're left with 2 pyruvate molecules, right? And we wanna break it down even further. So we're gonna take NADH. I should rephrase. We wanna cycle the electron carriers, because if you're gonna run out for when you break down more glucose, want to make more ATP. So what we do is we take 2 pyruvates, we stick those, or those things are gonna dump electrons on there, right? Booyah, become oxidized, NAD plus, right? That's oxidized, and that's gonna come back. And now you have this compound called lactic acid. You have 2 of them, right? And that's our final product for lactic acid fermentation. Moving on, let's look at alcohol fermentation. This is a little different. It's quite the same, but you'll see the difference here. So with alcohol fermentation, we go through glycolysis the same. The out-of-investment phase using the 2 ATP, blah, blah, blah. We said this a million times, right? We split it into the 2 G3Ps. We're going to take more ADP, right? 4 of them. We're going to come back. We're going to reduce into ATP. Right, and then we have NAD plus. NAD plus reduces itself into NADH. Quite simple. And now we have 2 pyruvates. Same thing as lactic acid fermentation and glycolysis in general, right? But here's where things get a little interesting, right? That pyruvate, instead of being just being more energized by that NADH, what first will happen is we're going to take this enzyme called pyruvate decarboxylase, right? Decarboxylase removing a carbon. That's how I like to think about that name, right? So we're going to take pyruvate. Just note, for simplicity, I'm going to just show that these are being broken down simultaneously, but they're going to be done in not non-simultaneous. Like one will go in the active site, react, then the other one will come in. But just for simplicity, let's just show it like this, right? And what's going to happen is we're going to break the carbons up, right? We're going to get 2 CO2s, right? So let cover the half of the screen here. Remember, we had that 3 carbon pyruvate. One of the carbons is broken down into CO2. The other 2 make this compound called acetaldehyde, I believe is what you call it, right? You have 2 pyruvates here, so you have 2 of them and you get 2 CO2s. Quite simple, but it's not ethanol yet. Ethanol is what we call alcohol, right? But we're at this weird compound. So what's going to happen, right? You need to keep cycling NADH, NAD plus, that electron carrier. So what we're going to do is we're going to take NADH, we're going to dump some electrons, and then those electron carriers will become oxidized. Now you have NAD plus. NAD plus gets sent off to the side, and we're left with 2 ethanol. It should look familiar. That's what we call alcohol, right? And that's why your bread will rise, right? Or not really. This kind of fermentation is why your bread will rise, right? Because of that CO2. That's why, I don't know, wine over time, you don't open wine for a very, very long time. When, like, let's say you put the ingredients to make wine, you cap it, right? A lot of CO2 will be produced in that bottle over time because it's fermenting. It's doing alcohol fermentation. It's taking that glucose, breaking it down into ethanol, and releasing CO2. That's why when you pop the cork off a wine glass or off a wine bottle, it's like, holy crap, that just went flying 5 feet in the air. That's how that works. Anyways, guys, that's all for now. Adios.